All right, so we should get started. So hi, I'm Maggie Flanagan. Thanks for joining our digital pathology webinar series today. I am assistant professor and Neuropath core leader at the Northwestern University Alzheimer's Disease Research Center. First, I'd just like to start with a disclaimer that this event is not sponsored by Halo, nor is NAC endorsing Halo through hosting and publishing this webinar. The goal of this webinar is for members of the Digital Pathology Working Group and our speakers to share their personal experience with using the Halo software to educate others on the software. So today we're talking about Halo image analysis software, and it is comprised of different quantitative digital pathology modules that can help simplify the workflow with your analyses. Today we will be having Dr. C. Dirk Keene, Associate Professor of Laboratory Medicine and Pathology, and the Nancy and Buster Alvord Endowed Chair and Director of UW Neuropathology, as well as Associate Director and Precision Neuropathology Core Leader at the University of Washington Alzheimer's Disease Research Center, with Jeanelle Azira Torres, a Keene Laboratory Research Scientist at the University of Washington, starting us off with the presentation, which will be followed by a question and answer session with the panelists. So take it away.
Okay, thanks, Maggie. And hi, everybody. I'm going to share my screen, and hopefully this works like it did in the practice. So is that showing up?
Yep, you're good.
Excellent. So I'll be quick because Jeanelle from our group is really the expert here. But what I wanted to do was just to introduce how we sort of came to searching for different applications for quantitative neuropath. We have been trying to transition into more quantitative approaches that help inform a lot of different molecular studies that we have with respect to brain aging, neurodegeneration, especially with our Alzheimer's Center, and of course, brain injury. And so this is just the mission of our Neuropath core. I think it's probably fairly similar to everyone else's Neuropath cores. The focus I want to show is our innovative approaches, right? That's always been a foundation for our Neuropath core, and we have focused for the last decade or so on, really emphasizing the attempt to quantify pathology. We've done that in a number of different ways through solution phase, protein extraction type approaches, through different biochemical approaches, and of course, different molecular approaches. But as you all know, most of what we have access to is in formalin fixed paraffin embedded tissue. That's where most of the banked material is around the country. And so the most applicable approaches are going to be using FFPE tissue. And so with that said, we have a number of different studies that we support. This is some of them. Of course, the Alzheimer's Center is of prime importance. The Adult Changes in Thought study, which is just renewed as U19, is of course a major focus, and our work with the Allen Institute in normal brain and in Alzheimer's disease is really critical. And so these quantitative approaches are necessary for us to really push the limits and understand what's happening in brain aging and dementia. And so this is our sampling protocol that we use just as a standard. We've developed a sort of modular approach. So this is your typical, over here is your typical neurodegenerative disease sampling that most of us do. We also have different sampling focused on CTE and TBI, and then of course a very large component that is based on structural and functional imaging. This is a slide just highlighting the sampling we're doing for our default mode and other functional connectivity networks, and this is really thanks in large part to Brian Edlow at MGH, Tom Grabowski at the UW ADRC, Christine McDonald here at UW, Kristen Dams-O'Connor, and others. The point is, we're taking a lot of samples. These are all in FFPE tissue, and we really need some way to manage this large amount of tissue, not just from a diagnostic perspective, but also from a quantitative perspective. And then with the renewal of the ACT study, with ACT-I, with some other grants, we are diving more and more deeply into, AI, machine learning / sort of deep learning approaches for neuropath, focused both on the imaging that we're doing on all these brains, but also on the histology. And so that is where we are basically at. And as of a few years ago, we started thinking, how do we take advantage of this, and how do we push this sort of limits? And so we recognized two real needs. One was this sort of efficient, relatively high throughput platform or approach to build quantitative neuropathology workflow and data. And then, of course, a mechanism to share images within our group and with our collaborators. And so with that, I asked Jeanelle from our lab to start to investigate. And she's done, as you'll see, a really deep dive into the different options and settled on Halo for our group, which really meets these needs, I think, perfectly. So with that, I'm going to turn it over to Jeanelle, and she can give an overview of Halo, and we can answer any questions that people have as we go over at the end. So with that, I will stop sharing if I can. Okay, Jeanelle.
Thank you. Good morning, everyone. I'm going to share my screen right now. Are you guys seeing my screen?
Yeah.
Okay. So my experience in image analysis is a little bit wide throughout the whole several years of investigation, but recently it's been focused on the whole slide image analysis using Halo image analysis platform. I don't know why it's sorry. I don't know why it's not working.
Frozen?
Yeah, like everything in technology has to be frozen.
I do have your presentation. I can share my screen that you sent.
Let me see if
you
I
can't get it.
can. I think it got better. Yeah, right now I got the control.
Okay.
Sorry, everybody. So right now, the most important part of our job is the relevance, and pathology has become relevant recently, not only because of the history that provides a lot of information, but because it enters in the digital analysis. So it uses the image analysis as a great potential to extract and quantify features in great detail in comparison with the assessment that the pathologist does. The quantification of immunohistochemical staining is one of the examples where automated methods are already being incorporated in research, in diagnostics, in teaching, and it's becoming more and more relevant with the use of artificial intelligence. So there are several technical requirements that we were mentioned in webinars, in past webinars, where we use the whole slide image, and we acquire that by the scanners. And also we use the image display for using a software analysis of the virtual slide that is located in server that is locally on your on-prem, or you can use cloud-based servers. So what is exactly image analysis? So this is a special discipline in digital pathology, that aims diagnostically important information in a very unbiased and reproducible manner by measuring, counting, and generating a large amount of specific data. So it's extremely complicated task. And if you do it right, you can get a lot of measure of morphological characteristics of tissue and cells, nuclei, and other organelles. Also, you can get counting of the cells of tissue components and the cytometry and pattern recognitions by measures cytochemical and histochemical and molecular cell or tissue features. So there are factors that you have to consider before doing the image analysis. Selecting the amount of sample that you wanted to do for your study, determining the region of interest, selecting the proper algorithms, and using the right type of annotations to determine and also determine the type of data you wanted to get. Keep in mind that also you need not only to determine those factors, but you have to get a very specific quality of tissue to be analyzed. Those pre-analytical considerations such as tissue harvesting, fixation time, embedding, cutting, standardizing the chemical staining, the type of auto staining that you use, or if you use a manual staining, those things can affect your image analysis. And also, if you add the complexity of having multiplex proteins, target proteins in your tissue, you have to also keep in mind the right combination of proteins, the host, the host of the antibodies, if it's polyclonal, monoclonal. If you use, for example, in enzymatic reactions, the type of the colors that you use for your enzymatic reactions to avoid the cross-reactivity issues, the antigen retrieval protocols that expose both antigens, they select the right sequence of antibodies to avoid masking of one antibody over the other one, the right incubation times, the patience, the auto-stainer, to simplify everything in your image analysis. This is our current pipeline. We get the slides from our pathologists and we scan them. We upload those images in our storage database in AWS. And we then, when we are about to analyze them, we transfer those images to FSX, which is a file storage directory system. And it helps us to connect with Halo. And we do the quality control with Halo. And then when the pathologists, neuropathologists approve the stain and what we are seeing in Halo is correct, then we do the image analysis. So for us, Halo has been a very pleasant experience. So Halo is a product from Indica Labs, and they specialize in quantitative pathology. They have several products, and I'm going to talk about two of them today. I'm going to give a brief introduction of Halo. Halo is an image analysis software that comes in modules. It's easy to use, and it has several add-ons depending on the needs of your study. It has a morphological and multiplex expression data on a cell-by-cell data basis. It's compatible with all image formats, the non-generic ones like JPEG, TIFF, Nikon, and the generic ones, for example, Nikon, Olympus, the Perkin-Elmer images, the Apereo images, size like Ventana, Deacon, Philips, several other ones. Sorry. Another product that they have is Halo Link. Halo Link is the web-based image analysis, slide management that takes the ability on basic tools from the image software and allows you to secure, share your images, share your analysis, and access them in a very secure way at any time from anywhere. The other part of the other product that they offer is Halo AI, which uses deep learning classifier add-ons, neural networks or artificial intelligence that can be trained to quantify tissue classes to segment the tissue for analysis with different modules. And the other product, which is more clinical and case-centric, digital pathology, is Halo AP. That allows you case review, sign-out, collaboration, and quantitative analysis, to the analysis in AI, et cetera, et cetera. There are 30 modules in Halo. You can have them for bright field or for fluorescence, immunohistochemistry stains, and also you can get whatever is right for your study. So in our lab, we got only 4 modules. which are the multiplex IHC that quantifies expression of up to 5 IHC markers in the nuclei, cytoplasma, or membrane, gives you the area quantification and the cell-by-cell or object data. The object localization, count objects, and stain intensity in each object, can measure 1 or 2 objects and stain in different chromogens, and quantify the object localization when it's applicable. The microglia measures the activation and detects the microglia. Some processes give you the reports of activating, inactivating microglia in compare with the non-microglia cells detected. It gives you the report of the processes and area and length, and also the process branches. The tissue classifier identifies multiple tissues using a learning by example approach that can be used in conjunction with other modules like fluorescent and bright field to select specific tissue classes for further analysis. So also, not only we are doing the analysis in the conventional way in digital pathology, but we are also enter in the artificial intelligence field. What is artificial intelligence is the leverages the computers and machines to mimic the problem solving and decision-making capabilities of human mind. One subfield is machine learning, is the concept that a computer program can learn and adapt to new data without the human intervention, identifies data and build predictions around that data. Another subfield of the artificial intelligence, which is a subfield of machine learning, is deep learning, which comprise neural networks. Differs from machine learning in how each algorithm learns. Also, the deep learning is an automation of extraction pieces of process by layers, has networks capable for learning and unsupervised from data, and utilizes a hierarchical level of artificial networks. And this is the one that it has been mostly used in our image analysis. So it works like this. So you train your algorithm. It will be segmented in small pieces. You train according to the classes that you wanted to classify. The computer divided in several tiles. It will be ranked, and then once you have trained it enough, he can see exactly the region that you already use as a training in different unseen whole slide images. So Halo AI uses those neural networks, and they have created different products according to the needs of the several investigations. So the masks that can classify the tissue, the specialty, the ones that are more focused on the nuclei and the phenotype of the nuclei segmentation, and they have created another one that helps you with the QC and remove those unwanted spots or areas that are not quite good for your image analysis, but that can interfere with your results. So now it's time to see in action. So I'm going to show you briefly one of the products, which is our window to our collaborators, is called Halo Link. And you can see it's very simple. It's a very simple interface. It can be organized depending on your study's needs. And you can add, for example, new study. For example, I can call it demo. And it's easy to open and create new studies with your collaborators. Every collaborator can access, depending on the credential, as a viewer, annotator, editor, or owner for that specific study. And that specific user have different powers. So for example, in my case, I can, for example, open the image. If I have the power of owner or annotator, I can do analysis in a simple way with Halo Link. I can, for example, check my image. I can see if any annotation has been made. I can see if any analysis have been made. And also I can run a specific analysis on this one. I'm going to show you. For example, like I wanted to analyze, it will show you the algorithms that are related to that specific study. And you can say, I wanted to analyze that specific image. And it will start doing the analysis. Other useful tool that a HaloLink has are the trace. With the trace, you can separate the software automatically copy those slides that you are interested in share with a specific group of people and separate it in trace. So when you do that, you are able to start seeing those images and start commenting those images. So for example, in this box, in the dialog box, I can comment, ask questions, ask suggestions, and use the annotations or the analysis to interact in a specific and organized way with my collaborators. So as you can see, the analysis is being doing, and I can also open the images in Halo if I have the access to it. And you can see the Halo interface. This is the interface, the viewer interface, where you can see the navigation menu here, the tabs here, the study pane, the image pane, and you can see that the analysis that I started in HaloLink is working perfectly well here in Halo. You can see the image tabs here, and each image opens with the label here and with the thumbnail here. So you can navigate your image and zoom in, zoom out, depending using your zoom toolbar. And you can also navigate using the thumbnail. As you can see, depending if you zoom in or zoom out, there is a gray transparent box here that indicates where exactly you are. Once the image is open, you can start doing the annotations. So you can go to the annotations tab, and you can start, for example, doing the annotations with this very simple toolbar that is in this corner, bottom right corner, and you can pan around using the pan tool. You can start annotating with the brush tool, or which helps you to create annotations, but also to modify the existing ones. You can also annotate by hand, whatever is your region of interest. You can exclude areas from that specific annotation. For example, if you are interested in this part of the tissue, but you are seeing, for example, a big chunk of tissue that is not correct or is interfering with your analysis, you can exclude that. Or, for example, if I wanted to annotate this specific area here, but I want to analyze this part and not this one. So I can invert the annotation, and it will be counted as my layers here, but it won't be counted in my analysis. So also you can use the magnetic tool, which is pretty useful for separating, for doing the tracings in a very specific edges of the tissue. So for example, you can start doing, and while it's active, it will start showing a dark blue line. And when it's inactive, it will immediately go with the color of your annotation. If you wanted to, for example, follow the pattern, you don't need to have a steady hand. So you can use the pixels edges and it combines the right pixels in the right way. So if you want to stop doing the annotation, you just click on the key bar and it will stop doing the annotation. The flawed annotation is pretty useful, for example. So it will separate or do the annotations with these pixels with the similar RGB chromogen. So you can do in a click a simple annotation. And for example, you can also have the ability to do the square annotation, the rectangle annotation, or if you want a specific square annotation, you just click the shift button and you can get a perfect square every single time. Also with this ellipse, you can have an annotation, elliptical annotation, or a perfect circle annotation if you desire to do that. You can also do the measurement, a quick measurement of whatever is your region of interest using the ruler. It will give you a specific annotation, or you can mark a specific cell or whatever is important to you, and you can add it in your annotation also. Also, you can add comments to your annotations, like for example, please check this cell and leave it for your collaborators or for yourself later, and you will show up specifically how many comments do you have for it and what specific annotation do you want it to see the comment about. Also in the toolbar, you can get the this icon that shows the histogram and the RGB values. For example, if you wanted to do a specific separation between gray matter and white matter by hand, so you can do it modifying the RGB exposure here, or you can restore the defaults. You can save that and use it for all the studies that you want later. There are other very useful tools that are in the advanced menu. So you can right click with your mouse and you can copy and move this, for example, to another specific image that is similar. For example, in a serial section, you have this specific tissue. You can copy this annotation and paste it in the other region. You can also move it if you made any mistake, or you can grow it. Oops, sorry. Grow the annotation or shrink the annotation depending on your needs. You can also have this advanced annotation. For example, if I am interested in knowing this specific blood vessel diameter, and I wanted to see how thick the walls of this blood vessel is. So I can start doing the annotation with my pen tool. Sorry, it's not precise right now. And you can have the ability to know the layer thickness. Oh, sorry, forgot to select both. And you can see here the options, the intervals for those lines to get an average of the lines that you wanted to get. If you wanted to smooth those lines, you can get another very reliable information, and you can see that every single one has a different layer and different information. So you can get an average of this specific layer thickness. Another useful tool in the advanced menu is, for example, you are interested in this specific region, but you wanted not to sample everything, but you wanted just a specific amount of data. So you can part this specific annotation in tiles, and you can, for example, reduce it by 80%. You can modify the microns you wanted to do it, and you can resample, and you can get a very random information from that one. Other useful thing that you can use with your annotation is the concentric regions. So in the concentric regions, you can generate, as the name indicates, concentric regions inward, outward of your region of interest, around, and you can modify the zones of the numbers of zones that you wanted to create. And once you create those ones, you can have a different layer, and each one will have different separated results when you do the analysis. So I'm gonna clear this view right now. Everything on the menu, you can also change the layers here. For example, if you wanted to rename it, you can rename the layer, you can change the color of your layer, you can import the annotations from another study, you can export the specific annotation, and you can delete it. It's pretty simple, straightforward. So once you have defined your annotation, you can do the analysis. And Halo comes with a very specific templates for analysis depending on the modules that you use. So in our case, it will give you the specific annotations here for, annotation algorithms for the error quantification, the color deconvolution, multiplex IHC, object localization, or you can save your own based on the parameters that are important for you, and you can start running, for example, the analysis in that specific annotation. So you can, when you are doing the analysis, you can start having a real time tuning of that algorithm for you. So you can make sure that you are doing the right modifications of the algorithm. So you can select one or several studies to do the annotation, to do the analysis, and you can start running the analysis. And once the analysis is starting, you can see here when the software is running the analysis throughout the annotation. You can navigate away. You can close this window, and you can see that this one that I started earlier in Halo link is showing you the results for area quantification analysis in my entire slide. You can toggle on and off those sections here, and you can see if it's accurate or not, that a specific analysis. You can navigate and you can get, and you can see the results here. For this specific analysis, you can get that summary results in the summary pane. You can see the tissue area, the specific area for new and positive according to my parameters. What is the percentage of area for that specific analysis? And in the case of my, let's see, analysis, I can get, for example, in the multiplex IHC module, I only can get, not only can get the percentage of area of my positive staining, but also the total cells that I demarcated as a positive according to my parameters, and I can get the object by object data. So you can see here, if I navigate in one by one, it will show me one by one the cells that are marked as a positive according to my parameters. So you can see, and you can get very precise data in a cell by cell basis. Also, there are also very important tools. You can export these results, the summary results in CSV file and then transform it to Excel file. You can get a summary of everything that you analyze in your specific tissue. Other useful tools that you have with Halo are, for example, the serial analysis a method. So you can go to the tools, you can go to your registration, and you can see what images are registered. It will ask you for those specific images. I have already analyzed this one. And as you can see, there are two images that are similar here. One is marked with ATA, and the other one with a tau4 antibody, and both are synchronized in navigation. So if I start zooming in here, the other one will show up, and I can compare side by side the two types of markers in every single tissue. And also I can have the possibility, if, for example, I'm interested in that one specific region, if I annotate this one in this side, it will automatically show in the other side. If you are not like quite convinced on the synchronization menu, you can, for example, start registering and adding landmarks. For example, if you want it to be extremely precise, so you can start adding those landmarks in that specific region, in that specific tissue. So you can say, oh, I want this as a landmark for my synchronization here, and this one. And you can say, apply those landmarks, and it will start rotating and trying to match the tissues according to the landmarks you are already adding. You can say, okay, and you can have a bit much more precise synchronization. So other useful tool that I found in the in Halo is the figure maker. So in the figure maker, you can just do a figure depending on your needs. You can add rows and columns, and you can add a border. You can add a scale bar, and you can you can add a border here. You can add labels in alpha or numeric way, or alphanumeric way. You can capture also the label if you want it. If you don't want it, that's okay. And if you are interested in that, you just lock it and you can open another image, for example, and go here. And for example, you are interested in this specific area, you can zoom in. It will zoom in with you, and you can close it, and you can see that is your figure here. So I think that's pretty much it of the general tools for Halo. So I'm open to your questions right now.
Perfect timing. I was just getting some questions coming through. So thank you so much. That was wonderful, particularly how you walked us through everything in the actual software. I thought that was really great. So I just wanted to introduce the additional panelists quickly. So we have Dr. Russ Huber also joining us today. He's an assistant professor of neurology at Boston University and the director of the PTSD Brain Bank, as well as Alyssa Myers, who is an application scientist from Halo Indica Labs, and she specifically works with our group. So to start off here, I have a couple of questions. The first one is, can you speak about Halo functionality for QC, for instance, for checking false positives and false negatives?
One thing that we use when we do the image analysis is to make sure that the pre-analytical parameters that we have to fill. We work in collaboration, very close collaboration with the neuropathologists. So they are the experts in determining what is exactly a positive or a negative. So once you have those very precise immunos or parameters for doing the image analysis, you can train your algorithm to avoid the backgrounds, for example. The functions, for example, are a huge problem for us because it gives you like a light brown color similar to the DAB, but the Indica labs have done an amazing job doing the algorithm sets and the parameters that you can avoid those backgrounds and to give you like a very close to perfection indication of what is positive and what is negative. And with the AI, you can get an even higher accuracy.
That's great, thanks. And this next question is also something I wonder about. How do the algorithms deal with staining like if it's homogeneous within the same tissue and across different slides? So for example, same staining protocol, multiple different slides within the case. How do you find that to be when working with Halo? These are common problems in quantitative path softwares where it tends to work well for samples from experimental models, but then fails when using postmortem human brain tissue.
So I don't understand quite well your question. So
yeah, I'll try to re-explain. So for other types of softwares, it tends to be the case that if you stain multiple slides, for example, from a mouse model brain, that will work well. But in contrast, for many softwares and people's experience, mine included, what may happen is that when you're working with human brain tissue, either multiple different autopsy brains or even one brain with multiple regions and the same staining protocol, how do you find the algorithm to deal with the staining?
In my experience with Halo, we have brains that have been stored for more than a decade. And those tissues have like this, their own specific high background. So in that way, the key is in the pre-analytical parameters, to standardize those pre-analytical parameters. Try to use exactly the same scanner when you do the image analysis with the image scanning. You try to use the same autostainer, the same protocol to avoid those variables. But you can see if I, for example, do the multiplex IHC cell by cell, I can do a QC very quickly of all my standard parameters, and I can see that it's being very consistent with Halo. I have found it very pleasant because we handle tissue, very old tissue with its own different parameters back then. And right now we are having a very standard protocols for extraction, for harvesting, for everything. And we find that I can use the same algorithm to analyze all those old cases and this one, and I have very consistent results.
If I could just add, I think that's a super important issue. Part of it is the old adage garbage in, garbage out, right? And so we can't, so Jeanelle's in the middle of a study right now going back into our archives for TDP-43. We have cases that are 25 or more years old. And I've been impressed with the ability of the Halo software to distinguish between, background, hemosiderin, other things that are sort of similar colors and what we're actually targeting. And there is this variation in staining, as you guys know, but it's all about going back and at the outset, taking and trying to train the system to get the system to recognize the variations from the different kinds of archived specimens or the variations. And so it's all that QC up front, which Jeanelle was mentioning, right? The neuropathologists are really more actively involved at the beginning, where we try to get cases that are going to span the different variations that we're going to see across the stains and the pathologies, to try to help make sure with this QC where you can go in and actually see what halo is masking as far as what it's counting as positive in these sort of test sections, to try to make sure that you're not over or under counting things or labeling things. And I'm with Jeanelle. I've been kind of impressed at the ability for the software to really pick up on these little tiny inclusions that we see with TDP across decades of different cases, like 800 cases. And so far it's been reasonably tight, but it's all about the QC up front. Mm-hmm. As Jeanelle emphasized at the beginning and again a minute ago, right? Get your staining working as best as you can, get the background as clean as you can, and then make sure that the images that are coming off are all, you got to make sure that the images that are being scanned are of appropriate quality. And the other thing I was going to mention is everything that Jeanelle showed is at 20x. We're scanning at 20x, not 40x, and it's sufficient to do these TDP43 inclusions, believe it or not. And so it's been really really helpful. But high quality up front, get everything as good as you can, automate as much as you can so that you don't have a lot of variation, and then things will, I think, work out pretty well usually.
Mm-hmm. Thanks.
I also wanted to add to that just for a second. We've been doing a lot of immunofluorescence work. And so the problems that you're talking about with IHC are magnified when you do immunofluorescence. And so one of the things, again, I'm just reiterating, it really is before you actually do the scanning that makes most of the difference here. Some people do normalization processes, and some people do some after correction for batch effects, but really it's getting your staining very uniform. And I think that's the most important part. Again, Halo itself is very good at distinguishing these things. I think a lot of times probably obviously better than by doing it by eye.
Mm-hmm. A few more questions. We have a lot coming in. So these two are somewhat related. So to what extent is Halo AI customizable? I feel convolutional models are not one size fits all, meaning we usually need a lot of specific training to have it detect our specific purposes. As I understand, Halo offers 3 pre-trained models. How well do these perform? Have you tried them on detecting plaques? And combined with this, does Halo AI allow a user to incorporate their own segmentation algorithms that were developed outside the system? Alternatively, can a user import their own annotations?
I've been working with the AI just over a month, and I've been very pleased with because most of the image analysis software are focused on the cancer research needs. But I have been found very pleasantly that I can modify those convolutional networks, and I can do my own mask depending on my needs. So I've been working with tau stainings, NUN, 6E10 stains in different regions of the brain to do the classification of the tissue, and I've been very pleased doing the modifications, the standardizations. You have to know exactly how to train the software, but they give you like a template, but it gives you also the flexibility to modify those templates. So I've been working pretty fine with all our needs covered so far, using the Halo AI masks.
And does anybody else have further comments on how interchangeable Halo is with outside algorithms?
Yes, I can speak to that as well. We do offer an interaction with our API, GraphQL you can use. Just to harp on a couple of things that Jeanelle mentioned, you do have the ability to feed annotations that exist already on your images into the Halo AI algorithms to basically have the AI algorithm feed examples into itself. You have a few AI algorithms that also come with sort of an out-of-the-box solution. So we actually provide 6 different neural networks on our Halo AI add-on product. A few of them have that out-of-the-box solution, such as our nuclei segmenter algorithm, as well as our nuclei phenotyper algorithm. You do also have the ability to create your own segmenter network or your own phenotyping network, and our Halo AI algorithms, whether they are the out-of-the-box solution or a customized solution that you've created as the end user, can then be fed further into your different analysis algorithms, like your RNA scope algorithms or your multiplex IHC algorithms.
Thank you.
Just one other quick thing. You can use annotations from eSlide Manager, which is the Aperio software. So I think that's kind of an important one since almost everybody uses Aperio scanners these days.
Yeah, very helpful. Thank you for that information. Another question, is anyone using Halo to quantitatively evaluate white matter pathology and or vascular pathology in neurodegenerative disease? I find these to be pretty subjective features and could benefit from some AI deep learning evaluation.
So I can start. We have a couple grants that are focused on white matter in vascular dementia and AD, and it's a chronic problem, right? And so how do you deal with that? We're just starting. I'm sure Jeanelle may have additional comments on what she's done, but you know, some of the stuff is easy, right? Astrocytes, microglia, the vessels that are in the white matter, oligos and oligo precursor markers, right? Those kinds of things are relatively straightforward because they're discrete entities. When you get into, you know, myelin pallor, we do LFBs with everything, and some of those kinds of things, I think it gets a little bit tricky. I think there's a lot of room for us all to sort of grow and learn and how better to assess the, you know, white matter injury, not just in neurodegenerative disease, but in trauma. Trauma is another big one, right? And so we're sort of working toward it. I think we have a long way to go, but you know, you can get intensity measurements from the software, right? It gives you a lot of different options to assess the stains. The issue again is if you're consistently doing the same staining as it's coming in, then you know, any kind of comparisons are relevant, but different aged blocks, different staining days or protocols, it all adds to the variabilities. I don't know if Jeanelle, if you have.
I just found that, for example, one of the key things that has Halo is the color deconvolution is very accurate. So you can separate the colors of your stainings. And for example, if we use LFB to measure the intensities in white matter, I just recently made a pilot study for a grant that was approved, and we got beautiful results analyzing the LFB intensity because the color deconvolution was extremely good. And we can see the differences in that gamma of staining in the white matter. And also we measure some blood vessels and the separation between the wall of the blood vessels within the white matter. So we have been working on that too. And it has been pleasant to work with Halo in that matter.
We've in our lab we've been looking at Luxol fast blue and then quantifying that, and it works pretty well. Like everyone else has said, the one thing that one's another one that can be very I guess dependent on the staining conditions. So again, definitely have to look on the QC on those to make sure that you're staining the same. But it actually works really well, and we just do sort of an area analysis on that. And quantify the intensity. So that works really well.
Thank you. So the next question is, For AI, does Halo, oh, okay, the question just disappeared. So I guess we'll go to the next question. Very nice presentation. If I understand correctly, UW is using 4 Halo modules currently to support / automate what a human would normally quantitate.
That is correct. We are using 4 because we found with those core 4 modules, we have more than enough. We have a lot of data. And in the next webinar, we are going to explain how to combine that those 4 modules to get most of the data in neuro, and also in the combination with the AI to get a much more precise extraction of that data. But we found that with those 4 modules is more than enough.
Yeah, if I would just add, humans would never be able to do the quantitation that Halo is doing. I mean, what humans would have done, like with ImageJ or something, would be to go and identify the different regions and then try to get the percent area and frequency of things, which is sort of the standard that we were doing a while back. But generally, for a lot of this, we never quantify anything. We just express either a really sort of semi-quantitative or ordinal value, like a Brock stage or something like that, or describe it as mild, moderate, severe, your typical pathologist stuff. So we aren't really quantifying things. We don't count much. Some other groups count things. You get all the counts that you could want, plus a lot of other measurements that with this and other applications that that quantify digital images. So it's not just Halo, but it's working really well. And like I said, we're on the side of a more high throughput type of an approach with a lot of cases. For that reason, Halo is really, as you saw with Jeanelle, it's got all these applications, and in theory, all of that can be taught and automated.
Thanks. I found the question that I lost. So for AI modules specifically, does Halo also provide ROC, AUC, and precision / recall values for the training validation sets? So Alyssa commented in the chat and said, we do provide a validation workflow for Halo AI, but I was wondering if anyone else may be able to just comment on what that looks like in a little bit more detail.
So it's actually a new feature that we're rolling out for our next release of Halo version 3.3, which should be out this month. So we do have a few little tutorial videos on it on our learning portal on our website, indicalab.com. Feel free, anybody, if you'd like to talk about it more, to reach out to us. It's a little bit of a hard concept to explain in a few minutes. So again, I'm happy to explain it, at a later date.
Okay, so it's not quite released yet.
Right.
Okay, thank you for the clarification. All right, and then time for one last question. How does Halo quantification correlate with your prior semi-quantitative Gestalt approach? Is this for primary diagnosis?
Right now, we, for example, I can give you a clear example with the TDP-43 staining that Dirk mentioned. We had to analyze the one pathologist is doing the analysis by eye with the computer, with the microscope, and I in the other side, I'm doing the analysis with Halo. And both we came with the conclusion that for example, the inclusion detection for certain cases using the eyes is like you can miss some cases because you can not see everything clear, especially with these tiny inclusions. But with the computer and with the object by object analysis, I could get like a precision of 5, for example, 5 inclusions that were missed with the pathologist as a negative presence of this, and I could see those 5 and we can compare, and the pathologist says, yes, those inclusions are correct. So some things that you cannot see with your eyes are being picked by the software if you train it really well and you do the QC really well.
If I could just clarify, we can see the inclusions once the computer tells us where they're at, but these are very, very small. And so we're trying to understand what the opportunities are here. Certainly as a screen to detect whether pathology is present or not, the HALO has been wonderful, especially when you're talking about trying to do TDP-43 in multiple brain regions across 800 people, right? So that you can go right to the HALO data and say, these are the cases that definitely have, that it's picked up some pathology. We can go verify those histologically and save an enormous amount of time.
Sounds pretty good. Sounds like maybe even better than the old way of doing things. So yeah, that wraps up the session for today. I just wanted to thank you all. Thank you, Doctors Keene and Huber and Jeanelle, so much for joining and showing us your experience so far. And I just wanted to announce that the next webinar session will be August 9th at 11 a.m. Eastern, and it is the follow-up to this session also on HALO, going into a bit more detail regarding some of the modules and AI. So see you there. Thank you.
Bye.
Thanks, everybody.
Thanks. Goodbye.
Thank you.